Good morning, students. So children, in the last video we have discussed about the restriction enzymes of recombinant DNA technology. Today we are going to talk about the next important tool for recombinant DNA technology, that is cloning vector. Children, these are the DNA molecules that can carry a foreign DNA segment into the host cell. So a cloning vector is a small piece of DNA taken from any organism into which a foreign DNA fragment can be inserted for the cloning purpose. Some of the important vectors can be used in recombinant DNA technology are plasmid and bacteriophages. Talking about the plasmids, so they are autonomously replicating circular extrachromosomal DNA. On the other hand, talking about the bacteriophages, so these are the virus infected bacteria and they are used as vector because of high copy number of their genome within the bacterial cell. Oh, here a new important term, copy number, is there. What is this copy number? Copy number is defined as the number of copies of vector present in a cell. It generally varies from 1 to 100 copies per cell. Children, talking about the plasmids have only 1 or 2 copies per cell, whereas in other their number can even go so higher in other forms. If a foreign piece of DNA is linked with the plasmid or bacteriophage's DNA, its number can be multiplied and become equal to the copy number of plasmid or the bacteriophages. Now there are some features that are required to facilitate cloning into a vector. The very first one is the origin of replication. Second one is the selectable marker. And third one is the cloning site. Let us see what they are. Talking about the origin of replication. So this is a sequence from where replication starts. And any piece of DNA when linked to this sequence can mate to replicate within the host body. This sequence is also responsible for controlling the copy number of a linked DNA. Origin of replication DNA की वो sequence है जहाँ से replication start होता है. And any piece of DNA when linked to this sequence can be made to replicate within the host cell, right? So this site is known as origin of replication, and it is abbreviated as ORI. Second is the selectable marker. Children, it helps in identifying or selecting transformants and eliminating non-transformants and selectively permit the growth of transformant. Transformant को identify करना, non-transformant को eliminate करना और transformation की procedure को पूरा करना. That is done by the selectable marker. Now children, selection of recombinants formed can be done by one of the following method, right? Like the very first one is inactivation of antibiotics. If a foreign DNA is ligated at the BAM-H1, right? Example लेके हम यहाँ बात कर रहे हैं, like if the ligation of foreign DNA at BAM-H1 site of tetracycline resistant gene in vector PBR332. So this is the plasmid PBR332. Now the recombinant plasmid will lose tetracycline resistance, but it can still be selected out from non-recombinant one by plating transformant of ampicillin containing medium. The transformant growing on medium containing ampicillin and then transferred on a medium containing tetracycline. Right? The recombinant will grow on ampicillin containing medium, but not on the containing tetracycline. On the other hand side, non-recombinant will grow on both the media, and thus recombinants are selected. Right? So in this example, one antibiotic resistant gene help in selecting the transformant, whereas the other antibiotic resistant gene get inactivated due to the insertion of alien DNA or the foreign DNA and help in selection of recombinant. Right? So in that way, the recombinants are selected. It is a cumbersome procedure. Therefore, the alternative selectable markers have been developed with differentiated recombinant and non-recombinants on the basis of their ability to produce color in the presence of chromogenic substrate. Right? Now, second is the insertion inactivation. The second method is the insertion inactivation. The recombinant and the non-recombinant can also be differentiated on the basis of color production in the presence of chromogenic substrate. Here, a recombinant DNA is inserted within the coding sequence of an enzyme beta-galactosidase, which result into inactivation of enzyme. Therefore, the bacterial colonies have inserted plasmids show no coloration, while those without plasmids show blue color. So in that way, recombinant and non-recombinant are further categorized or differentiated. Now children, vectors for cloning gene in plants and animals. In plants, the tumor inducing plasmid of Agrobacterium tumefaciens is used as a cloning vector. Right? It delivers a piece of DNA known as tDNA in the Ti plasmid, which transform a normal plant cell into tumor cells to produce chemical against pathogen. Similarly, retrovirus and adenovirus, papillomavirus are also now used as cloning vector in animals because of their ability to transform normal cell into cancerous cell. Right? So these are some of the vectors. Now children, let us talk about the third important category is the cloning site. These are generally required to link the foreign DNA with the vector. वेक्टर हमने पढ़ लिया, फॉरेन डीएनए हमने पढ़ लिया, अब क्लोनिंग साइट क्या है जहाँ से वो जुड़ता है, अटैच होता है. फॉर दिस वेक्टर रिक्वायर्स वेरी फ्यू और सिंगल रिकॉग्निशन साइट्स फॉर कॉमनली यूज्ड restriction enzyme. If more than one restriction site is present within the vector, it will generate several fragments that will lead to more complication in gene cloning. The ligation of the foreign DNA is carried out by the restriction site present in one of the two antibiotic resistant genes. So in that way, the new or the foreign DNA is being ligated to the plasmid. Talking about another very important tool, that is competent host. DNA is a hydrophilic molecule, so it cannot pass through the cell membrane. In order to force bacteria to take up the plasmid, the bacterial cell must first to be made competent to take up a DNA. So the competency is the ability of the cell to take up a foreign DNA. Now let us talk about the methods for the transformation of DNA. The very first one is the chemical method. Children, in this, the cell are treated with a specific concentration of a divalent cations such as calcium, which increase the efficiency with which DNA enters into the bacterium through the pores on its cell wall. Right? So the cell are incubated with the recombinant DNA on ice, which is followed by placing them briefly on the 42 degrees centigrade and then putting back into the ice. So this treatment enables the bacteria to take up the recombinant DNA, and that method is known as heat shock method. एक बार ice, एक बार 42 डिग्री सेंटीग्रेड, अगेन देन आइस, राइट? तो हीट शॉक ट्रीटमेंट इसे कहा जाता है. सेकेंड मेथड इज द फिजिकल मेथड. इन दिस मेथड द इंट्रोड्यूस फॉरेन DNA into host cell is done by microinjection. In this method, recombinant DNA is directly injected into the nucleus of animal cell, right? तो microinjection. इसी तरीके से एक मेथड है बायोलिस्टिक और जीन गन. So this is another method suitable for introduction of DNA into plants. क्या होता है इसमें? In which cells are bombarded with high velocity microparticles of gold or tungsten coating with DNA. So in that way they forcibly enter into the bacterial cell. In this picture, it shows DNA is injected into the nucleus of host by the process of microinjection, right? Another very important tool is the bioreactor. Children, talking about the bioreactors. So bioreactors are the large volume vessels, approximately 100 to 1000 liter, in which raw materials are biologically converted into a specific product. Individual enzymes using microbial plants in animals or human cells. A bioreactor provides the optimal condition for achieving the desired product by providing optimum growth condition like temperature, pH, substrate, salt, vitamins, and oxygen. The cells can also be multiplied in a continuous culture system. In this, the use of medium is drained out from one side while the fresh medium is added from the other side to maintain the cell in their physiologically most active phase. This type of culturing method produces a large biomass leading a high yield or the desired protein. Do you know what are the components of bioreactor? First one is the agitator system. Second one, an oxygen delivering system. Then a foam control system. Then a temperature control system, pH control system, and a sampling port to withdraw culture periodically. There are two types of bioreactors. The most commonly used bioreactors are stirring type. Stirring type bioreactors are further of two types, simple and staggered. In simple, staggered tank bioreactor is usually cylindrical or with a curved base to facilitate the mixing of reactor content. And the staggered state tank bioreactor also facilitate the mixing of component and ensure oxygen availability throughout the bioreactor. Alternatively, air is bubbled through the reactor. Talking about the very important thing, that is by downstream processing. After completion of the biosynthetic phase, the product has to be subjected through a series of processes before it is ready for marketing as a finished product. The process includes separation, purification of product, which is collectively called as downstream processing. Finally, the product is then subjected to quality control testing and kept for the suitable preservatives. Downstream processing and quality control tests generate differ from product to product. And finally, the product is released into the market. Right? So bioreactor is what? It is the very important tool which gives the products in a very large amount. Right? In the next video, we will further discuss about the recombinant DNA technology. Till then, have a nice day and thank you.